Hello, CACNA1A community. This is Ping Hong Fox, the Science Engagement Director for the CACNA1A Foundation, and I'm really excited to tell all of you that the Foundation has awarded a $50,000 research grant to Dr. Sam Young at the University of Iowa. So Dr. Young is a professor and the Vice Chair for Research in the Department of Anatomy and Cell Biology. He is also a professor and director of the molecular auditory research in the Department of Otolaryngology, as well as the University of Iowa Carver College of Medicine Distinguished Scholar Award recipient. Fun fact, he was also a starting defensive tackle for the 1995 Princeton University Tigers Ivy League championship team back in his undergrad days, and he remains a huge fan of the sport. And we're actually Big Ten rivals now because he's an Iowa Hawkeye and I'm a Wisconsin Badger, but that we'll let that pass. So one focus of Dr. Young's lab is investigating how synaptic transmission, or how neurons talk to each other, is regulated by calcium ion channels. And part of this research involves developing molecular tools called viral vectors that enable the study of this process in neurons. So I'd like to welcome Dr. Young to the CACNA1A community and to our research network. Thank you so much for being here today to talk about your work with CACNA1A and how it will impact those living with CACNA1A related disorders.
Well, first of all, I want to thank you for the opportunity, and I want to thank the CACNA1A Foundation for the grant award. We've been really overwhelmed and really excited about this. And on a personal level, it's a far cry from the days of being on the football field at Palmer Stadium in New Jersey to now in the research lab. But I think if anything, the struggles on the football field in terms of a team effort with one focus, one team, one vision, I think is exemplified in this team of the CACNA1A Foundation, basically to try to cure these CACNA1A disorders. And I think that's really what we're most excited about and something that we're really interested in going after.
Right. So, yeah, we're definitely happy to have you on our team.
Yeah, I can't speak to the Big Ten rivalry, so we can discuss this at Camp Randall or
yes,
in the
absolutely. All right. So before we get into the details of your project, we do want to just give a little bit of background to our community about the basic concept or science of gene therapy. So could you just briefly explain what gene replacement therapy is?
Yeah, sure. I mean, gene therapy as a concept has been around for maybe 70, 80 years. It's this idea, once they discovered genes, that you have a gene that doesn't function correctly, that you could introduce the gene, a good copy of that gene, to basically restore the function. So to put it in more basic terms is that genes, for the most part, encode proteins. Okay, and that these genes encode these proteins to do certain jobs within the cell. Okay, and what happens is that you get mutations in these genes, some good, some don't do anything, but some of them do bad things to the protein. And so essentially that protein doesn't function anymore. And that misfunction of the protein can result in bad things to happen to these cells so they don't function. And then this basically results in essentially this dysfunction. All these genetic disorders are basically caused by issues with the genes not encoding the proper protein. So yeah. The viral vector part, right, to get into that is then the big question is, what the gene is, what needs to be done, but how do you replace it? Okay. So in the 80s and in the 90s, right, it was known that viruses over millions of years of evolution can do a very good job of getting into cells and delivering genes, right? That's what viruses do, okay? What viruses also do very well is make you sick and do bad things, right? So in the late 80s and 90s, a lot of these virologists, and one of them I had the fortune of working with, Jude Samolsky, who I did my PhD work at UNC Chapel Hill, was to basically take out the bad viral genes, okay? And basically that way you don't have any viral genes in there, and then you put in whatever gene you want, but then utilize all the good stuff that the virus knows how to get in a cell. So now you basically have a delivery truck, right? That can now essentially go where we want, right? Or a drone now that we're in the early 2020s now. But essentially now what you have is you've taken all the good parts of the virus, you use it as a delivery method to basically use that to get the gene that you want into the cell.
All right. I know that we often have CACNA1A families coming to the foundation and asking if, gene replacement therapy is even possible for our community. And they're always asking, what, if any, is the status of this research for CACNA1A related disorders? So could you explain why this approach hasn't really been feasible or possible for this community yet?
Of course. I mean, it's a big issue, right? So just to set the table in the landscape of what's going on is that right now, gene therapy has been very successful in curing certain monogenetic disorders, right? And the issue there is that's largely been built on the back of AAV gene therapy, okay? AAV, for those of that you don't know, is a small virus, right? That's been successfully used in the clinic, but it can only package roughly about 4.5 kilobases of DNA. So just to put it in there, it's in this case, essentially, it has a very limited ability to carry genes, okay? So it's limited on what it can carry. It does an excellent job of getting into cells that you want, but it can't carry much, okay? Now, the CACNA1A gene itself, okay, is roughly 2 times bigger than the packaging capacity of AAV. So in this case, to treat the fundamental root cause of CACNA1A disorders, you cannot use AAV to do this, okay? And so that's put up a major roadblock. And so for the gene therapy regards, not that, to say in this regards, people have now focused on, well, if we can't treat the root cause of CACNA1A disorders, maybe we can treat some of the other things downstream of the CACNA1A disorder with another gene therapy strategy to get at a different pathway. Well, those look to be somewhat ameliorate some of the CACNA1A disorders in some cases in these preclinical trials. It doesn't treat the root cause of the disorder. So that's an issue. Now, what you need then, okay, is, well, we need a viral vector, okay, that can carry the CACNA1A gene. And what I also didn't mention, all the regulatory elements, right, that allow for proper expression. Because one of the things in gene therapy that we need to be aware of is that we also exist in this Goldilocks syndrome, Goldilocks world, where you don't want too much, you don't want too little, you need it to be just right within a range, right? And so you need a lot of the regulatory elements to make sure the right amount of protein is being made. And then the other issue in this regards is that a lot of disorders express a mutant protein, right? And you need to get rid of that mutant protein and replace it. So then you have another issue, okay? So now the ability, you have to carry a lot more that's even bigger, maybe 3 to 4 times bigger than the AAV packaging capacity. So a virus that I work on is called adenovirus, okay? Adenovirus is it's a well-known human virus. It's been studied for decades, right? For at least 70, 80 years. And a lot of RNA splicing, a lot of what we know about cell biology, a lot of what we know about the causes of cancer biology are due to working with adenovirus, right? And so it's a very well understood virus on what it does, okay? And some of these, they're used really well for vaccine development in humans. One of the issues with adenovirus is essentially that can it get into the right cell type that you need it to? Okay, so it does get into certain cell types, but when we look in the brain, been known for 30 years, if you use the most common serotype of AD that's been used in preclinical trials, it's a serotype called AD serotype 5, it does not get into the cells that you need it to get into. So if we look at essentially that roadblock, right, that you have a virus, adenovirus, adenoviral vectors, they could carry this, but they can't get into the cell types, okay? So that's a problem. And then, the issue is solving, is it safe? What is the long-term toxicity issue? So a bunch of other groups in the late 90s, early 2000s, vectorize adenoviral vectors for something called helper-dependent adenoviral vectors. Now, these helper-dependent adenoviral vectors can carry up to 36 kilobases of cDNA, about 7 times to 8 times more than AAV. But regardless, those vectors still, with all their tremendous potential, still don't have the ability to get into the right cell type. So essentially, that type of research has been very stagnant for the last 20 years of using HDAD to treat certain neurological disorders. And so I think that's been one of the issues on why it hasn't been possible to the community for date. The other thing that it cannot be estimated is AAV has been wildly successful, right? I mean, it's been wildly successful. So there's been a massive investment of pharma, right, into AAV gene therapy. And while pharma is great, right, they've done an amazing amount of stuff. They're very good at late stage development. What needs to be done is a lot of early stage development right now. And so pharma has gone after AAVs, and they have not really gone after and invested very heavily into the development of helper-dependent adenoviral vectors for use in neurological disorders. And that's okay, right? That's how fields come and move. I mean, 30 years ago, you couldn't get anybody to invest any money into AAV research. So things come and go, but I think that's where it's been stagnant. And in terms of CACNA1A disorders, right, the big issue is figuring out, can you get into those right cell types? And so then the other issues, can you express the gene? Do you have someone that knows what CACNA1A does, right? And also has essentially a background in virology. Well, I know one person that does. That would be me, right? So you need a basically, not to show for myself, but essentially what you need is a combination of expertise, a multidisciplinary approach to solve this issue. And so I think that's why things have been relatively stagnant in this regards into the gene therapy.
Great. Thanks, Dr. Young. So yeah, we have a large gene. None of the current technology none of the current vectors have the capacity to really carry it into the brain safely and effectively. And so let's talk about your project then, right? So could you provide a lay summary of the research that we're funding and how you envision this work moving our community closer to viable gene therapy treatments?
Of course, of course. So what we've been focusing on is basically trying to solve the #1 issue in gene therapy. So for your audience, the #1 issue in gene therapy is delivery. The #2 issue in gene therapy is delivery. And the #3 issue in gene therapy is delivery. And if you solve 1, 2, and 3, then you get to go work on #4, which is repeat administration. Okay. So we've been trying to tackle the problems 1, 2, and 3 on delivery. Okay. So what we wanted to do is basically, we knew that the most common type of ad vector, ad type 5, does not transduce the cell type called Purkinje cell. Okay. Now, we've been heavily focused after cerebellar ataxias in this regards, and those are largely due to CACNA1A dysfunction. Okay? And these cell types, which are the sole, provide the sole input to the DCN, okay, to provide essentially output in this regard. Those cells degenerate over time. Now, the current ad vectors can't get into Purkinje cells. Okay. Now, we know from our previous research, we've done a lot of studies in different areas where we know we can deliver essentially the CACNA1A gene to really wild type levels to basically cure, to restore function in mutant backgrounds. But we couldn't get into these Purkinje cells. So we made a bet, right? And this basically, I mean, I do want to thank the University of Iowa for this Distinguished Scholar Award, which basically allowed me to do this high-risk project where I took a lot of this money and basically, actually, I took all the money and basically said, we're going to solve this problem. Okay. It was a completely unfunded project, completely high risk. We didn't know if it was going to work. So our goal was essentially to say, okay, we just don't want to deliver this gene to a mouse, because who cares, right? You need to deliver it eventually. The end point is to go into a human. So what we did was we used a humanized mouse model, which contains receptors, humanized receptors that the adenoviral vectors normally use in humans, right? And then what we did is we made a different, what we call chimeras of these viruses, that we altered the tropism of the virus, okay? So now essentially what we did is we made about 10 to 20 delivery trucks that had the potential to go to all these different spaces. So my postdoc who's working on the project right now, Dr. Emra Kuhl, together with a graduate student, Chechi Okorafor, and then we have and other individuals in my laboratory, they started screening these vectors to see whether or not we can get these viral vectors into Purkinje cells. So of course, you already know the answer to this story. The answer is we can, right? So we knocked down the first major roadblock. So 30 years ago, it can't be done. You can't get ad vectors into Purkinje cells. As of 2023, we can get them into Purkinje cells. This is a major advancement, okay? So now the fact that we can get into these Purkinje cells, now what we need to be able to figure out is how many Purkinje cells can we get into? And we need to work out the regulatory elements so we make sure we're producing the right amount of the gene in the cells that we can. And then now we have essentially a way to build preclinical models in preclinical mouse models to show, can we cure the ataxia, right? Can we cure, a whole host of other CACNA1A disorders? Each one may have their own little trick that may be needed to essentially get to that. But our play is that we know what the cerebellum does. We know what the Purkinje does, Purkinje cells are doing, and we know if we can rescue them, then we could treat a whole class of CACNA1A disorders. And so that's what the research is funding right now. We're basically, the research that we propose and what it's funding is improve upon the delivery, because now we solved problem number one, but now we got to solve problem number two, which is again, delivery, because now we need to figure out how many cells do we need to deliver these viruses to stop, let's say, the degeneration or effectively halt the progression of these diseases, right? Our goal at this time point is, our ultimate goal is to cure the disease. But if we can just ameliorate some of by stopping the progression to improve the patient's life, right, at certain levels, then we start building towards that ultimate cure. So that's what the research is funding now, and it is continue to develop these vectors now to improve and characterize their tropism. We're going to basically we're developing the correct transgene cassettes to basically make sure it's just the right amount, and then show that we can do this in a mouse model to cure the cerebellar ataxia. Then once we have that done, then this opens, even for bigger projects, which is we now have humanized mouse models of, CACNA1A conditions, and then to just start broadly testing.
Right. Thank you, Dr. Young. So you've basically developed these vectors or delivery trucks that can bring CACNA1A into these Purkinje cells. And just a reminder to our community that CACNA1A is expressed all over the brain, but it's really highly expressed in the Purkinje cells. So it's definitely playing a big role in that. And so targeting those cells for, as a starting point for treatments is a great place to start. And Dr. Young is able to get these vectors into those Purkinje cells. And I mean, this is essentially a precursor to, future gene replacement therapy, but it's a precursor, and it's setting some of the groundwork to get there at some point. So thanks. So then just, let us know then, what are you most excited about in regards to this project and working with the community and the foundation?
I mean, I'm really excited about the project because I really want to say that the real heroes in this type of research, right, It's not the scientists, okay? It's really the patients and the patients' families, right? That are at the forefront, that are the advocates that are pushing for this, and that are eventually going to be signing up to try these treatments to do this, right? Those are the heroes of gene therapy. And I really want to say that, up front, right? And the excitement is for us is that we are committed essentially to developing a therapy that cures all CACNA1A disorders, right? And so for us, we showed that we can do this, right? We took a problem that was not solved for 30 years, right? Like, you can't get into Purkinje cells. It doesn't work. It doesn't work. And at the end of the day is, why doesn't it work, right? And so sometimes in science, right, you just have to think about the problem and say, why not me? Why shouldn't we solve it? And so we solved it. Now, a lot of these ideas that we've had before, like, oh, we could do this, they're possible. And basically what I'm really excited about working with the CACNA1A Foundation is, one, is the team, the leadership team, the foundation team has the right vision, right? They are basically focused on funding the science, and not just funding science in regards of we'll fund some. They're really targeted in going after things that are going to be innovative and basically develop into potential cures, right? And because a lot of these things, like I said before, pharma is very great at late stage development. But what pharma's weakness is at early stage development. So a lot of times, if you follow a lot of foundations, especially for DMD, they were essentially basically the force behind this that got a lot of these gene therapy trials going. And so you have to rely on a leadership team and the patients families, the advocacy to say, this is important. We need to fund this research, and we need to fund research at a preclinical level, right? To basically show that this can be done. The other thing is that, not one thing will cure, right? I mean, hopefully we get that, but it's the understanding that you're going to fund multiple approaches to basically try to cure the disease or fix the patient's lives and to basically improve their lives over time. And then working with the foundation and the community is going to be fantastic, because you have experts from all across the spectrum, from clinicians to basic researchers to patient advocacy groups, that basically dial you in to basically understand, okay, this is the underlying issue. And you stay focused in that regard, because a lot of times in basic research, it's wonderful, but just because it works in a lab doesn't mean it's going to work in the clinic. And so it basically focuses you on, we must solve this issue. This is what our endpoint is. And so you're really attacking that. And I think that for us basically motivates us every day, is that this is a disease, we think we have it. And that when we wake up and to do these things, we're super excited that we know our research is basically going there to try to help improve patients' lives and improve the lives of the patients' families.
Oh, we definitely appreciate that. Not just the foundation, but the community as well. And as I've said, we at the foundation are really excited to be funding this work and seeing where it goes. So this is wonderful for the CACNA1A community. Dr. Young, welcome to the team. We're honored to have you on it. Thank you again for being here today to talk about your work, for the work that you are doing and telling us about, how it's going to impact our communities. So we really can't wait to hear updates about this project from you.
Well, thank you again. Thank you so much. And thank you to the CACNA1A Foundation for believing in our work. And, you're going to get everything we got. I left it all on the football field many, many years ago, and you're going to get that same type of delivery. We're going to leave it all out there, and we're going to give you everything we got to deliver this cure across the goal line.